Love to see where everybody's at. Welcome, California. Welcome, Malaya. So we got somebody.
Good
Wow, coming in fast. Can't hardly read it. Great
evening,
to see if we
America.
have people from maybe different time zones this time, since we're a little more favorable to our friends in Asia and our
Yeah,
friends in
Hong Kong.
Australia, New Zealand. Welcome, Canada, Nebraska, Austin, Texas,
Singapore, welcome.
British Columbia.
Florida.
Lord, Peter, you got some more Malaya folks coming in. So, bienvenido, Colombia.
Colombia.
Hello, Seattle.
Texas.
Welcome, everybody, to the Tree Fund webinar series. We'll be starting off here in about 2 minutes, as is our tradition. Find that chat function. Let us know where you're coming from. Hope everybody, depending on what time zone you're all, are either having a good afternoon, a nice evening, or a nice morning for those of you in the Southern Hemisphere on the opposite side of the world.
Another Malaysia friend.
Yeah.
I saw
Welcome.
Sydney and I thought that was Australia, but there's a Sydney, Ohio. Didn't know that. Welcome, Kauai. Beautiful spot.
California, Washington State.
Wow. Well, folks will continue to trickle in. Heath, we might want to hit that record button. I think we're about a few seconds out from the top of the hour.
We are.
Welcome, Georgia. Georgia in the house. There we go. Somebody from Alabama. Whoop, whoop. Welcome, Birmingham. I probably missed somebody else. Well, everybody, it is 7 p.m. Central Standard Time here in the southeastern United States, anyway. My name is Bo Broadbeck with Auburn University, and I want to welcome you to the Tree Fund webinar series, to our first webinar of 2026. So, Happy New Year. I know it's a little late to be saying that, but hey, it's the first time I get to speak to all of our Tree Fund webinar attendees, which is awesome to be back for another year with this webinar series. It's always a pleasure. As y'all may have noticed, we're trying a new time. Our speaker, Peter, is going to be joining us from Malaya, and he's with the University of Malaya, so it's tomorrow morning for him over there. So a little more favorable time, and we're exploring this new time for this webinar. So welcome on behalf of Auburn University. It's always a pleasure to be a host of this webinar series. And now I'd like to introduce my co-captain on this webinar series, Heath Hupke, to talk to us a little bit about Tree Fund.
Thank you, Bo. And again, I echo our New Year's welcome to everyone. So thank you for kicking off the Tree Fund webinar series here in February. And thank you for, again, attending tonight, today, tomorrow morning, wherever, and I guess whenever you are. And for those who may be here for the first time, or maybe you're still learning about Tree Fund and our programs, we are the Tree Research and Education Endowment Fund. We are a nonprofit or a non-governmental organization. We have endowments to support research and education in the urban forestry and arboriculture fields. And we operate our organization as we operate, we do rely on donations from partners and sponsors and individuals. So in December, for those that were able to join us in December of 2025, we had a matching challenge from our board of trustees. And so thank you all for those that were able to step up and donate $5 or $100. It was greatly appreciated. It helped Tree Fund immensely. So thank you all from us again. But we also want to thank our Crown and our Platinum partners here that are represented on this screen. And so with their generous help, we're able to continue to provide this webinar series for free. We're able to, for those that are attending, we'll get CEUs, and then we will have this posted online on our YouTube page afterwards. And so, thank you for that. And a big part of this reason that we have this webinar series, a part of the Tree Fund's mission is not just to fund the research, but to get this research into your hands, into fresh hands. And so, as you'll see today, this is freshly completed research, and so some of this information is going to be very much brand new for everyone. So if you want to be able to continue to support this, if you attended last year, or again, today's your first day and you want to support Tree Fund, our mission, and not just getting research funded, but also getting the word out and spreading the information into the hands of arborists, please consider following this link, making a donation, and supporting the work that we have going. And one of the wonderful things that's well represented today as we continue to talk is Tree Fund is very much an international supporting organization that looking at our 2025 webinar information, we had over 7,700 people attend. 5,000 of those were unique attendees with 58 countries represented and all 50 states. So usually we do this in America in the afternoon, and we're happy to be able to expand that time to better serve our presenter today, and to welcome you all around the world, whether you're in America or outside. So welcome and thank you. And as a reminder, your name and your certification number when you registered for this program, your ISA information CUs were already captured when you logged in. We know who you are, so you do not need to put in the chat your ISA information. If you are wanting a certificate of education, or if you have any questions from past webinars that you might be missing a CEU, please email me at education@treefund.org. That's going to be the best and the quickest way to get that. If there's anything you need, please let us know. And then we do have some things already lined up for the next coming months. So our March and April webinars, those registrations are live and able. So if you haven't registered for them, please do so. Those will be kind of at what we define as our normal time, which would be about 12 o'clock Central Standard Time. And that's going to be on March 3rd, Tuesday, March 3rd. And then we have another one on Tuesday, April 21st. And then a save the date for yourself. We do have a webinar lined up on Tuesday, July 21st as well, but that registration link is not quite available. It will be by our next webinar. Now, I'm happy to introduce one of our partners, especially for the Jack Kimmel International Grant, which was the funding grant for today's presentation. I'm going to introduce here Kathy Bentley. She is the co-chair and a grant coordinator with our friends, the Canadian Tree Fund. Kathy?
Thanks, Bo. Just say welcome, everyone. The Jack Kimmel grants provide needed funding for arboriculture and urban forestry researchers all over the world. And we have partnered with International Tree Fund. And so this grant, the International Jack Kimmel Grant, is partially funded by Canadian Tree Fund, and it's administered by International Tree Fund. And there's some participation from Canadian Tree Fund in the evaluation process. Usually we're awarding one grant a year, maximum of $10,000. And this started in the 90s, and it began with the Tour de Trees ride, because there's always Canadian riders, and this enabled Canadian Tree Fund to send money into international. And that was sort of the idea, was to extend our Jack Kimmel grant, which is a Canadian-based grant awarded across Canada and extend that into international avenues. So that was the beginning of that. And the name, it's named after Jack Kimmel. To honor him, he was a director of parks for City of Toronto. And that we remembered his contribution of 46 years of leadership to ISA and the Ontario chapter. So without further ado, I would like to say in 2023, the International Jack Kimmel Grant was awarded to Petr Hedenec in Malaysia. And he is going to be speaking today about his project. And it's already on the screen there. His project, he's with the University of Malaysia at Terengganu. And he's a soil ecologist there. His research focuses on soil biota and particularly fauna and microbiota and how these organisms interact with soil processes to influence broader ecosystem functioning. So through his work, he explores critical role soils play in maintaining healthy and resilient ecosystems. So congratulations on your grant and go ahead, Peter.
Okay. Can you see the screen now?
Yeah.
Okay.
Looks good. Thank you.
I thank you for opportunity to present my work at this webinar. Good evening for those who are in North America and Canada, and good morning for those in Malaysia, China, and Singapore, and Taiwan as well. So let's start. I will talk about how termites can affect soil properties and how these changes in soil properties could affect the tree growth. So let's start. First of all, some few words about my institution. So I started at the University of Malaysia, Terengganu since 2021. I'm originally from Czech Republic. So I moved from Europe to Terengganu to Malaysia. And this university is primarily focused on the marine science, but I'm sitting at the Institute of Tropical Biodiversity and Sustainable Development, and we are also doing research in terrestrial ecosystems like tropical rainforests, agriculture sites, urban parks, etc., etc. So at the beginning, few words about the history of urban parks. So the urban parks start in ancient civilizations. Early parks were de facto private gardens for royalty, like Babylon Hangings Garden. Then we jump to the 18th century, where the idea of public parks emerged in Europe influencing the landscape. The good example is James Park in London. Then in 19th century, due to urbanization, the urbanization led to overcrowding, and there was the prompt need to create some place for recreation. The good example is Central Park in New York. Then in 20th century, parks became center of public life, offering green spaces, spaces for relaxations, sports, and community events. And in 20th century to the present, there is the growing focus on sustainability, environmental benefits, and inclusivity in park designs. And here you can see the illustration of the hanging gardens in Babylon. Here is Central Park, and this is on the right side is the public park in Kuala Nerus in Malaysia. So the parks provide ecosystem benefits which are important for human being. First is the climate regulations, typically cooling effect, regulation of microclimate, and etc. Then parks in general can help to clean the air, the cleaning effect of the trees, where the microparticles are adhered to the leaves and it's and also producing of oxygen as well, and the catching of pollutants from the air. Then Other benefit is connected with water management, where the parks can promote water retention and flood mitigation. Parks in general are biodiversity hotspots. It's a home for rare species. Noise reduction, I would be. It's quite funny because in the park sometimes you can see some noisy events like concert and so on. Nowadays, it is going to the light carbon storage or effect of the tree species to the carbon storage through the different leaf litter, and also soil quality, prevention of the soil erosion and so on. On this picture, you can see the public park in Kota Kinabalu in Sabah. This is another part of Malaysia in Borneo. Trees are the essential part of the urban parks. By definition, simply definition is the trees are woody perennial plants with single stem and or trunk bearing lateral branches above ground level. Sometimes we also consider the minimum height, which is 1.3 meters. Trees are essential part of urban parks. They also providing benefits for human well-being, but now they are under extreme anthropogenic pressure. Same like urban parks, the trees have positive role on air quality improvement, energy savings, food supply, if we have orchards and so on. Also, it is good for recreation, to improve mental health and so on. Trees are important for the soil, and we have two main ways how trees can affect soil biota. So first, we have direct interactions, typically mycorrhizal fungi, root feeders, plant root pathogens. These are the interactions where the organisms directly interact with trees or with other plants in general. Then we have indirect interactions where the trees interact with soil biota through the leaf litter. And here we have two strategies. We have the trees which producing a leaf litter with high CN ratio. And this leaf litter with high CN ratio promote fungal energy channel with very slow nutrient turnover. In contrast, we have leaf litter with low CN ratio, which producing leaf litter with high quality, and this leaf litter support bacterial energy channel and with very fast nutrient turnover. So this is the picture showing a complex view on the trophic interactions in the soil. So you can imagine tree which producing different quality leaf litter. It supports fungivores or bacterivores. It supports bacteria or fungi. Fungi are consumed by fungivores like spring tiles and some oribatid mites like oribatid mites. Bacterivores are typically consumed also by some mites and also by protozoans and so on. And these organisms are consumed by higher organisms, and this creates the complex trophical or complex food webs. Back to the urban parks. Urban parks are under extreme anthropogenic pressure, typically increased concentration of pollutants and climate change. And this is hand in hand with pests and diseases. Here I give some example. In Northern Hemisphere, you know very well ash borer beetle, Agrilus planipenis. This is beetle native in Northeastern Asia, but became invasive in Europe. And this is destructive for ash Fraxinus, genus Fraxinus in general. As a fungal pathogen, we can see also in Northern Hemisphere ash dieback. This is the fungus also native in Asia, but rapidly spreading across Europe and North America. This is example. These are also invasive species, so they are not native they are not native in North America and Europe, but spreading because they have no natural enemies like We can also see the ash borer beetle in Asia, especially in the northeastern part of China. And this beetle is basically not so abundant in that area. This is example of plant pathogens in urban park of Singapore. Urban soils in general harboring diverse microbial communities, including higher proportion of pathogens. And this pathogen pose significant threat to the public park itself and also by human, because sometimes they can damage the trees, which can fall and harm the people. This is the disease of kaya tree with black ring on the trunk. This is caused by Felinus. Felinus is the pathogenic or wood rotting fungus. In Europe, we have Felinus robustus. It's another species. This is just how we can recognize, or we can recognize three types of wood rotting. First, we have soft rot fungi. These are typically slime molds, common molds. These are the initial rotting, typically on fresh wet wood. Then we have brown rot fungi. Brown rot because these fungi decompose cellulose, which is the white in the color, and therefore this wood became brown after the cellulose is consumed. And this because this is the fungi consume the cellulose, and the remaining material is lignin, which is brown. Typically, we have Glauphilum trabellum, Letiporus sulphureus. These are Letiporus sulphureus, especially is the wood rotting fungus growing on the trees in temperate Europe. Then we have white rot fungi. These are fungi decomposing lignin, and lignin is brown, therefore the remaining cellulose is more light. So this is why we call it white rot fungi. And typically Trametes versicolor or Phanerohet chrysosporium. These are the fungi typical for the white rotting. In tropical areas, the termites pose serious threats to the urban parks. Just a few words to introduce the termites. Termites are very abundant in tropical areas. By the way, they are also source of the methane because they have the methanogenic bacteria in the gut, some of them. And we have two main groups of termites. We have soil feeders. Typical soil feeding species is the Pericapritermes. This one, this guy with skewed mandibles. And we have wood feeders, typically Globitermes or Macrotermes. Soil feeders, they are responsible, or to the certain extent also the wood feeders. These are also responsible for bioturbation. Bioturbation is the vertical transfer of the nutrients in the soil, and they are also responsible for formation of soil aggregates because they consume also the wood feeding termites. They consume wood, but also with the remainings of the soil. And if this material passes through the gut and defecate, it is very rich in the bacteria, and it is important in formation of soil aggregates. This is a picture of Macrotermes. This is the most one of the most abundant termite in Malaysia. They are wood feeding, but not really, because they use wood to mix the wood material with saliva and soil, and they are creating this brain-like structure, the blue structures, which are the fungal gardens. So they grow the fungal termitomycete fungi, and they feed with these fungi their workers and soldiers. So this is here is example the distribution of soil organisms across the globe. What is important in temperate zone, the most abundant soil animals are endforms and microbial feeders and omnivores. But in tropics and subtropics, termites are dominant soil organisms in tropical and subtropical areas. And what is important, in tropical areas, the termites are responsible roughly for the 50 and 70% of annual litterfall. So it means all litterfall fallen to the ground, 50 or 70% is consumed by termites and other organisms. In our areas in Terengganu, we found two most dominating species of termites. First was Macrotermes, and second Globitermes sulfureus. Macrotermes, it is characterized with large mandibles, and as I said before, they create the large termite mounds, and they are cultivating fungi on the mushroom gardens or fungal gardens. In contrast, the Globitermes sulfureus, they are smaller. They have color like sulfur, so this is like sulfuric yellow color, and they have not so big mandibles, and their defense mechanism is they produce the sticky substance. So their body is filled with the sticky substance, and if they are attacked by ants, they produce like glue substance, and it is defensive mechanism against ants. This is example from Pakistan, how the parks are infested in Pakistan. So they found this is the percentage of the number of infested trees. We can see here in this, we can see the high variability in termite infestation between the sites. But most vulnerable to the termites are the trees which were imported from another areas, like in canal site, it was the Populus euroamericana. In Pakistan, the most dominant termites were Odontotermes, Coptotermes, Microtermes, and Heterotermes. So these are most, these were the most dominant termites in Pakistan, in urban parks of Pakistan. This is from study of Afzal and Rasib from 2022. This is how termites can be also beneficial to a certain extent. Here in this study, the authors, this is a study from China, and the authors measured the differences in physicochemical properties across different sampling layers in active and abandoned mounds in comparison to adjacent soils. And termites nests showed the higher concentration of carbon, nitrogen, calcium, and nitrite anions, but a lower concentration of NH4, the ammonium. So, based on this, this inspired me to ask for the questions how the vicinity of the termite's nest to the tree can affect the tree performance and also the soil, not only soil physicochemical properties, but also the soil biota. We focus on fungi because in fungi we can find the pathogens and also another the beneficial fungi like ectomycorrhizal fungi, saprophages, and so on. So in our study, the main objectives were first to investigate whether distance from termite nest can affect growth performance of trees. Then to measure the soil properties at different termites nest and investigate also how is the microbial diversity and community composition of fungal community at the different distance from the termites nest. We hypothesized that termites nest will show higher concentration of carbon, and also it will change the soil texture. We also expect a higher diversity of fungal community in termites nest, and we also hypothesized that tree tree performance will be higher in close to the termite's nest. So the trees which are closer to the termite's nest will show the better growth performance. We choose two urban parks in Malaysia. First was Kuala Nerus, which is the public park dominated mostly by palms and also ficus and pterocarpus. And then we choose Taman Botani, which is the park in our university. This is park dominated by the local species like Dipterocarpus, Shorea, agatis, and Melaleuca cayuputi, which is the typical tree in the coastal areas of Terengganu. Yeah, in total, we obtained 72 samples. We did 2 sampling campaigns in May and June, and then we took the samples on the nest, 5 meters from nest and 10 meters from the nest, and we took 3 samples from each distance, and yeah. So for tree growth performance, we used digital dendrometers from EMS Brno. It is a company in Czech Republic. So this is the digital dendrometers which measure the growth. You can set up the intervals 5 minutes, 4 hours. It has quite long lifespan, battery around 3 years. Yeah, the resolution is at the micrometer scale, and yeah, the problem is the price. This is quite expensive. So, and we got in-kind help. So we got some the equipment from the company, but the problem was because the people are curious, what is it? And the problem is if you touch the equipment, then it is highly biased, the measurement. So this is the problem. So we move the measurement of the trees to the Tasik-Kenyer Geopark, which is the this is the more far from the human settlement, and it is also visited by humans, but not so often. And there are the places where we can cover this equipment, so we can measure for the longer period. So for the measurement of the trees in our urban parks, we used the standard caliper and then taped dendrometers. So as I said before, the soil samples were subjected to the classic physicochemical soil analysis. So we measured soil moisture using gravimetric methods. Then we measured pH and electrical conductivity using the electrode test, and also electric conductivity salinity. Then we measured the total organic carbon, total nitrogen, CN ratio using the CNHS analyzer. Then we measured also plant available phosphorus, and we measured also soil texture using the sedimentation method. We also extracted DNA from the soil. We used the Power Maxi soil, where we, in classic or in more common DNA kits, we used the microgram or milligrams. We used milligrams or grams, but this is to cover the larger proportion of the biodiversity. We used the Power Soil Max kit with 10 grams of soil. So we have quite nice nice imagining of how nice picture about diversity of fungi. We used ITS2, it is the biomarker for fungi, ITS2 transcriber. We prepared a sequencing library. We used Illumina sequencing the machine at the Chengdu Institute of Biology in China, and we used UNIT database. This is the common database for taxonomic affiliation of individual fungal classes and the genes. We used also fungal traits database for affiliation or assignation of the fungal functional groups. In terms of statistical analysis, we used the mixed linear model. Distance from nest location and termite species were considered as the fixed effect, and sampling time and location as a random effect variable. We can jump to the results. This is total organic carbon, and we can see significant differences between termite nests and different distances. So we can see in higher concentration of total organic carbon in termite nest. This is because termites contribute to the accumulation of and transformation of organic matter through the feeding activity, and they also accumulate plant residuals and organic rich material in the nest. So this can be a reason why we found higher concentration of organic carbon. We found higher salinity also in termite nest, but this can be because, as you can see in this picture, this is the termite nest of the macrotermes, and the termite nests are elevated. So the higher salinity or higher conductivity may be because it is elevated, so it means it is a bit drier than other parts, and therefore it has higher evaporation and higher concentration of the higher conductivity and higher salinity. We can see also differences in clay and silt. This is because termites, especially macrotermes, they have ability to select soil particles to improve the termite nest. So they used clay particles and silt as a building material. So this is why we can see the higher proportion of the clay and silt in the termite nest in comparison to the soil from a larger distance from the termite nest. This is the alpha diversity. This is on the y-axis. You can see ASV. ASV is amplicon sequence variance, and this is, we can consider, it is not really the species richness, but we can consider it as a proxy of the species richness. So we can see the higher species or higher ASV richness in the termite's nest in comparison to the soil from larger distance. Here we can hypothesize that termites nest create very specific chemical and microclimatic conditions that provide specific niche or specific microhabitats for the specialists, for the or specialized fungal species. This is a heat map showing how soil physicochemical properties can affect fungal diversity. Here we measure different alpha diversity indices. So we can see the moisture and pH negatively correlated, conductivity correlated positively, but this is rather because we found also larger differences in soil properties between sites. This is finally, here we can see the view on the, we can see the picture, how it looks at the species, how looks species composition. This is at the class level. We can see Eurothiomycetes, the blue one, these are the dominant everywhere. We can see statistical significant differences in Dottideomycetes and what is important, Agaromycetes, because we can see Agaromycetes are, or we found these fungi are statistically significantly higher in the termites nest. This is because in Agaricomycetes also include the specific fungi which are related to the termites nest. This is, we can see differences between various species, Globitermes and Macrotermes. We can also see the Agaromycetes Agaromycetes shows higher relative abundance in Macrotermes in soil associated with Macrotermes. We also analyzed different functional groups, and we can see the relative abundance of pathogens is lower in termites nest, but relative abundance of mycoparasites and animal parasites is larger in termites nest. This is because animal parasites, it could be also the fungi which are parasites or which are associated with the termites. So it can be the pathogens of termites. We also expect that it can be associated with presence of termites, high population of termites, higher individuals. It means higher abundance of animal parasites. We also expect that changes in soil properties, especially soil texture, like higher concentration or higher proportion of clay and silt, can be associated with the decline of the plant pathogens, because the plant pathogens, they need the plant tissue or some plant residuals as well, and these are not so common in the termites nest. I mean the living plants. We can see also differences between two species. We compared Macrotermes and Globitermes, the soils from the termites nest, and we can see litter saprotrophs are more abundant in Globitermes. Also, dung saprotrophs are show the increase in Globitermes. Yeah, we can hypothesize that this is connected with foraging activity and behavioral pattern between these two species, because the Globitermes, these are the foragers. They accumulate the wood material and other plant residuals in the termite nest, while the Macrotermes, they are cultivating the mushroom gardens, and they have quite different the feeding habits. This is the non-metric multidimensional scaling. This is the beta diversity. It means beta diversity indicates differences between various habitats. We found that the fungal community in termites nest was distinct from those at 5 and 10 meters. Communities associated with soils at 5 and 10 meters indicated the similarity, while the communities in the termites nest were more separated from those at 5 and 10 meters. Yeah, this is almost textbook example. This is biodiversity between these two species, and we can see the larger difference or larger separation between the Macrotermes and the Globitermes. Yeah, this can be also linked with different feeding strategies. Macrotermes are fungus growing, while Globitermes are wood feeding, active foragers. And yeah, we also measured indicator species. This is the heat map. The black color indicates the species with higher indicator values. So in this heat map are present only these ASV or amplicon sequence variants, which show indicator value larger than a certain threshold. So we can see termites nest indicated higher number of ASV with higher indicator values. Mostly we found, unfortunately, we did MySeq sequencing of ITS2, and this is not quite good for the higher taxonomic resolution. So we found, for example, many unidentified ASV. Mostly, we found the common species, what we can find in the soil, like Penicillium, Aspergillus, Aureobasidium, and so on. This is also same. This is similar picture where we compared Globitermes and Macrotermes. Both species host specific indicator values. So for I forgot to mention the measurement of the tree performance is still ongoing, because we finished 1 year already and we didn't find any differences between in the tree growth between the tree species. In terms of measurement of growth performance, the digital dendrometers are still ongoing in Dasik-Kenir, and we will also continue with DAP dendrometers in the parks to measure. But we need at least 3 years to get some picture, if any. But for now, it seems that there is no significant differences between the tree growth. Or also, we try to extract DNA from the leaves, from the tree leaves to indicate some pathogens, but it is also we failed to amplify the suitable amplicons for the markers. So in conclusions, termite mounding activity don't show any effect on growth performance for now. The termites mounding activity increase total organic carbon and conductivity in topsoil. Soil termites nest showed changes in soil texture as well. Termite mounds indicated higher alpha diversity of soil fungal community. Then beta diversity differed across different distance from termites nest. So it means it was separated between termite nest and the rest of the samples. Termite mounds or termites nest showed indicated lower relative abundance of plant pathogens, and also termite mounds indicated higher relative abundance of animal parasites. Also, the relative abundance of saprotrophs, different groups of saprotrophs differed between Macrotermes and Globitermes. So that's all. Thank you for your attention, and thank you for opportunity to present this work. And many thanks Tree Fund Jack Kimmel Foundation for financial support of our research. So thank you very much.
Thank you very much, Dr. Hedenec. Great presentation. Very fascinating topic, something I've never really considered or thought about, at least here in the United States. So really, really interesting. We do have a number of questions. For all of y'all joining in, please remember to put any questions you have for Dr. Hedenec into the Q&A, and then I'll get to those questions as time allows. So Dr. Hedenec, the first question I have here is somebody wrote, how long does a typical termite nest last, And do you think the lifespan of the nest has an impact on how significant the soil changes around it may be?
Thanks for this question. I think yes, because we tried in our study. We tried to have, if possible, the same nest. It was very easy in terms of globitermes, but macrotermes, yes. I think yes. The age of the nest can significantly affect the quality of the soil, especially for macrotermes, because they sometimes produce really large termites nest. Some in Africa we can see even more than 1 meter, 2 meters large, and yes, it can affect significantly the surrounding soil. It is interesting also for globitermes. They have quite small termite nest, but they have a large quantity, so they can reach even 10,000 individuals per termite nest, and also they have very large the foraging range. So yeah, can be.
And with that idea of a foraging range, underground, that there was a question here about, with the mounds, how large is the underground area? Is it contained to a certain area, or does it spread and have that effects on the soil in a broader area?
It can be. Yes, it can go to the larger, definitely larger part of the termite mounds is below ground in some species. So it is a bit problematic because if we want to measure this, we need to completely destroy all nests. So we need to dig around the nest, then like cup, turn away, and then dig in case of macrotermes, dig the fungal mushrooms, mushroom gardens, and then the queen chamber. So yes, the large part is below ground, and it can be quite large. It can be quite large area below ground.
Thank you. And there was another question about which tree species you were measuring as part of your study.
We measured. Where is it? We have here this. I cannot remember now. Where we go? Well, Melaleuca cayuputi. It is the typical species in Terengganu, and then I have window now. Yeah, here we can Dipterocarpus and Shorea, mostly Dipterocarpus shorea, and in Taman Kuala Nerus it was Dipterocarpus, and the palm species mostly. So for the Taman Kuala Nerus we measured Ficus and Pterocarpus. We tried to compare mostly the broadleaf species, so Ficus, Pterocarpus, and then Shorea and Dipterocarpus and Melaleuca in Taman Botany at UMT.
Thank you. And another interesting question here that one of our attendees asked us says, do you think your research is applicable, to termites in temperate countries?
Temperate countries, if I are only subtropic, so if we set up some biogeographical scale, then temperate, no temperate zone. Maybe in Europe, the most northern part where we can find termites is southern Austria. So for the temperate zone, it is not quite suitable.
Okay.
mostly tropics and subtropics.
That makes sense. And let me filter through the questions here. There's another good question. It says, will you be able to carry your study on long enough to find out whether the impacts on tree growth hold true during times of drought?
It is a good question. Definitely, we need at least, I would say, at least 5 years to get some nice picture about tree growth performance, especially with these digital dendrometers, because here we have the monsoon season between late October and early October, between October and late February. There is the monsoon season with very excessive rainfall. So after that, we can expect the significant growth, and there is also the dry season or dry period, and yes, it can have effect on this. It has.
Thank you.
So definitely we need larger time scale to measure, to get some significant picture on the growth performance.
And does Malaysia have a distinct wet and dry season, like some tropical parts of the world?
Yes, monsoon and non-monsoon season. There is every day raining, but we have the monsoon season, especially in Terengganu, where there is the really excessive, really significant raining between October and February, and then there is a bit drier period with less rainfall.
Thank you. Yeah. I was going to jump to another question here. It says somebody wrote that since biodiversity is generally considered beneficial for the health of an ecosystem and the urban forest, do you believe either of these species of termites is beneficial?
It depends. Definitely, for if you have a house nearby termite nest, it is not so beneficial. Like especially in case of Coptotermes and also Globitermes, they can also attack the human houses. The problem is when in plantations or in forest plantations or in oil palm plantations, it can be a problem, because when the termites are overpopulated, it can also they can also harm, or they can also attack the trees. But in general, they are, especially the soil feeding termites like Pericapritermes, and also, even though it is the wood feeding, but Macrotermes also, it has beneficial effect because of bioturbation, but it is how to say, if it is beneficial for the nature, yes, it is beneficial, because it consume the old tree branches. It helps return. It helps to release nutrients. So for the nature, it is beneficial. For human being, if you have a plantation with trees, or with oil palm, or with other rubber trees, then it can be problem. It can be problem, the overpopulation of termites.
It's that
Or in the parks, in parks, it is problem, because sometimes they can damage the trees, or they can harm the trees, and then it can be also for human, can be dangerous for human.
Yeah, it's one of those classic answers. It depends. It depends on where they're at. It depends on the scenario.
Yeah, exactly.
Lot of possible outcomes there and scenarios. I think I'll have time for one more question. Do termites? Somebody wrote that if asked if termites might increase mycorrhizal networks. did you see any evidence of that?
Not, but I believe so, because especially some foraging, hypothetically, yes, because some foraging termites, if they are running through the leaf litter and collect they can collect some small branch or leaf. So yes, they can contribute to the spreading of the ectomycorrhizal species or also arbuscular mycorrhiza. It is good for another research. good, good research question. Yes, I believe so. It can be a factor which contributes. In general, if you look at the decomposition, uh, in my previous work, back to my master thesis, we had the litter bags accessible and not accessible for different animals. And those which were accessible, they show the larger decomposition rate because they also introduce the specific fungal and bacterial decomposers. So I believe it can be also true for the mycorrhizal species.
Well, thank you. Well, thank you. And thank you very much for sharing your research today on the Tree Fund webinar series. I want to thank Kathy Bentley also for joining us and telling us a little bit about the Jack Kimmel Award and also having the Canadian Tree Fund fund this study. It's always fantastic to see different studies with different ecosystems, different parts of the world that all contribute to our better understanding of trees and urban environments. So thank you for sharing that. Thank you to all of you who are listening today, whether you're joining in the morning tomorrow or if it's in the evening. I know some of you in the Eastern time zone, it's getting a little late out there. But thanks for hanging on and joining us today or this evening on the Tree Fund webinar series. We will be back. Go to treefund.org, look at the webinars tab. We have another few webinars listed coming up. We're going to have a full schedule of webinars in 2026. So hope you're able to continue to join us as we continue with this fantastic webinar series. I just want to throw a quick reminder because we always get questions about CEUs. Just a reminder, when you registered, we captured your name, your certification number, and we will submit that on your behalf directly to ISA. Give us a little bit of time. Usually takes about 4 to 6 weeks to post. So with that, I want to sign off this evening. Thank you, Dr. Hedenec, for taking the time to join us. Thank you again, Kathy. And see y'all on the next Tree Fund webinar series.
Thank you.
And thanks, Heath.
Thank you. Bye.
Cheers.
Have a good night.